Hello students, welcome back to my channel. Today I am taking class for class 11th NCERT books CBSE board chapter 10 cell cycle and cell division exercises. This is your first question. What is the average cell cycle span of a mammalian cell? Your answer is here. The average cell cycle span for a mammalian cell, we can follow this step. Understanding the cell cycle. The cell cycle is a series of phases that a cell goes through as it grows and divides. It consists of two main phases, interphase and the mitotic phase. Phases of the cell cycle. Interphase, this is the growth phase where the cell prepares for division. It has three subphases. G1 phase, the cell grows and synthesizes proteins, last about 10 hours. S phase, DNA replication occurs. G2 phase, the cell continues to grow and prepares for mitosis. Mitotic phase, this is the phase where the cell divides. It includes karyokinesis, the division of the nucleus, which has 4 stages: prophase, metaphase, anaphase, telophase. Cytokinesis, the division of the cytoplasm resulting in 2 daughter cells. Duration of the cell cycle. For mammalian cells, the entire cell cycle takes 24 hours to complete. So the average cell cycle span of a mammalian cell is approximately 24 hours. It is up to you. If the question is asked in very short, you can go through the last steps duration as a what is the average cell cycle span of the mammalian cell. If it is asked in the detail, then you can go through these steps. Next, distinguish cytokinesis from karyokinesis. Now here you can see karyokinesis and cytokinesis. Under karyokinesis, nucleus of the cell is divided into 2 daughter nuclei. Under cytokinesis, cytoplasm of the parent cell is divided into 2 daughter cells. Second point, initial step in the cell division, final step in the cell division. Under karyokinesis, daughter chromosomes are divided into 2 daughter nuclei. Here, under cytokinesis, 2 daughter nuclei are divided into 2 daughter cells. Fourth point, under karyokinesis, genetic material is equally distributed. Here, cytoplasm and cellular organelles are equally distributed. Fifth point, a process involving in various steps for segregation of genetic material. Here, a comparatively simple process. Here, spindle formation and movement of chromosomes occur. Here, formation of cell plate or cleavage furrow occurs. 7. Sometimes followed by cytokinesis, does not occur without karyokinesis. So, these are the main points for karyokinesis and cytokinesis, right? Your third question, describe the events taking place during interphase. The events taking place, see, interphase is a period of the cell cycle for growth and DNA replication. Preparing the cell for division, it is divided into 3 phases. G1 phase, it is also called first gap, and first gap. Here, the cell grows and becomes metabolically active. It synthesizes protein and organelles necessary for the next phases. The cell prepares its DNA for replication. S phase or synthesis, the cell replicates its DNA, doubling the amount of DNA in the cell. Each chromosome is duplicated, resulting in 2 identical sister chromatids, but the chromosome number remains the same. G2 phase, that is the second gap, the cell continues to grow and prepare for mitosis. It synthesizes proteins and RNA required for the mitotic division. The cell checks for any DNA, also checks, CHEC, it is C, checks for any DNA damage to ensure proper replication. Your fourth question, What is G2 quiescent phase of the cell cycle? The D0 phase occurs when the cell cycle is inactivated because mitogens and substances high in energy are not available. At this stage, cells still have an active metabolism, but stop dividing means they do not grow or differentiate unless the organisms requires them to. Caudate nerve and heart cells are always in zero phase, G0 phase. Now, your next question is here. Why is mitosis called equational division? Here, mitosis is called equational division because the number of chromosomes in the two daughter cells is equal to the number of chromosomes in the parent cell. For example, if a parent cell has 18 chromosomes and two daughter cells are produced through mitosis, each will have 18 chromosomes. Your next question, name the stage of the cell cycle at which one of the following events occur. Chromosomes are moved to spindle equator. It is at the metaphase stage. Centrosome splits and chromatid separate. It is anaphase. Pairing between homologous chromosomes takes place, zygote. It is the meiosis first, prophase की substage है ये. Crossing over between homologous chromosomes takes place, packaging, right? Now the next, describe the following. Synapses, bivalent, chiasmata. This is here. Synapses, the physical pairing of homologous chromosomes where one chromosome from each parent come together. It happens during the zygote stage of anaphase 1 of meiosis. The pairing is aligning homologous genes along their length and is stabilized by the formation of a protein structure called the synaptonymal complex, right? Next is bivalent. The paired structure of two homologous chromosomes formed by the synapses. Each homologous chromosome has two sister chromatids. A bivalent contains a total of 4 chromatids. It is also called a tetrad because it consists of 4 chromatids. Bivalents are crucial for the exchange of genetic material through crossing over, which occurs between non-sister chromatids. Chiasmata, they are X-shaped structure formed between homologous chromosomes during prophase 1 of meiosis. They represent the point where crossing over or the exchange of genetic material occurs between non-sister chromatids, leading to genetic recombination and increased diversity in offspring. These structures are crucial for the proper separation of homologous chromosomes during meiosis. They are formed in the packaging stage of prophase 1. Here you can see, I can show you. See, this is the lactotin stage where the homologous chromosomes are there, they are separated. In zygote, the homologous chromosomes show pairing. This pairing of the homologous chromosome, this is called synapses. ये देखिए, ये homologous chromosomes pair कर रहे हैं. पैक्टिन में यही जो paired homochromes थे, जो ये bivalent है, यानी कि दो साइड से, ये देखिए टेट्राड बन गए. ये टेट्राड पैकेटिन में. अब यही डिप्लोटिन स्टेज में क्या होता है, ये जो टेट्राड्स हैं, ये टेट्राड्स, दो टेट्राड्स, ये आपस में एक्सचेंज ऑफ क्रोमोसोम किएस्मेटा, ये जो पॉइंट ऑफ इंटरेक्शन है, ये किएस्मेटा फॉर्मेशन करता है. अगर इसको आप डिटेल में देखेंगे तो आप ये देखिए, ये जो टेट्राड्स हैं, ये टेट्राड्स ये आपस में क्रॉसिंग ओवर करते हैं. इस क्रॉसिंग ओवर में जो इसके पार्ट्स जो हैं वो आपस में एक्सचेंज होते हैं. दैट इज द रिप्रेजेंटेशन कैपिटल एबीसी स्मॉल एबीसी. तो ये कैपिटल एबीसी जो ये है, ये इधर चला जाएगा और ये वाला जो इधर चला जाएगा. आई होप इट इज क्लियर. नाउ योर नेक्स्ट all the points. I hope you are able to get it. That is the synapses, bivalent, chiasmata. Draw a diagram to illustrate your answer. I have shown you the diagram. How does cytokinesis in plant cells differ from that in animal cell? Now here, see, cytokinesis in plant cell or cytokinesis in animal cell, how it differs, see, how it differentiate. First, in case of plant, a cell plate is formed from vesicles originating from the Golgi apparatus and endoplasmic reticulum, whereas in animal cell, the cytokinesis in case of animal cell, a contractile ring made of actin and myosin filament forms a cleavage furrow. Second point, the cell plate starts in the center of the cell. This cleavage furrow starts at the outer edge of the cell. Third point, it grows outwards in all directions towards the existing cell wall. It deepens inward or centripetally. The fourth point is here, the cell plate fuses with the plasma membrane and cell wall, creating a new cell wall that separates the two daughter cells. Whereas in case of cytokinesis in animal cell, the furrow continues to pinch the cell into eventually separating it completely into two daughter cells. I hope it is clear. Next, find examples where the four daughter cells from meiosis are equal in size and where they are found unequal in size. See, four equal sized daughter cells are produced during spermatogenesis, that is formation, that is in the sperm formation as seen in human males. Four daughter cells of unequal size are produced during oogenesis, that is egg formation, which results in one large ovum and three small polar bodies. An organism undergoes meiosis, but instead of all cells being equal, the cytoplasm is divided unequally. This results in one large ovum and contains most of the cytoplasm, and the three smaller polar bodies that are non-functional and eventually reabsorbed by the body. I hope it is clear. Now the 10th one, distinguish between anaphase of mitosis and anaphase 1 of meiosis. Now here you see anaphase of mitosis and anaphase 1 of meiosis. Sister chromatids separate at the centromere and move to opposite poles. Homologous chromosomes separate and move to opposite poles. Here the sister chromatids move and there the homologous chromosomes move across. Sister chromatids separate, remain attached to each other at the centromere. Two identical sets of single chromosomes, where here two sets of chromosomes, each consisting of two sister chromatids. Here, daughter cells are genetically identical to the parent cell. Here, daughter cells are genetically diverse due to crossing over in prophase 1. Now, the next one, distinguish, this you have in the anaphase, list the main differences between mitosis and meiosis. Now here you can see the main differences between meiosis and mitosis. Differences between mitosis and meiosis. Mitosis, the division takes place in somatic cells. As a result of this division, growth occurs. This takes place in reproductive cells. As a result of this, only one generation, this character passes from one generation to other. Here completed in one stage, they are completed in two stages. Here prophase is smaller as compared to the, here prophase is longer than prophase of mitosis and divided into 5 substages. No crossing over takes place, crossing over takes place. Synapses does not take place at metaphase. Here synapses between homologous chromosomes takes place, that is the bivalent stage. Here at metaphase, centromere is inwards is towards the equatorial plate and end of chromosome towards pole. Centromere divides. Here at metaphase 1, the centromere is towards the pole and ends of chromomere towards equatorial plate, but centromere does not divide. Here chromatids are long and thin, here chromatids are shorter and thick. Only this is sufficient for you, right? Now, here distinction matter, it's over. Main differences, mitosis, meiosis, we have done it. What is the significance of meiosis? Now here, significance of meiosis, you can see, under this production of gametes, meiosis is the process that creates gametes that are essential for sexual reproduction. Reduction of chromosome number, it reduces the chromosome number from a diploid twin to a haploid and state, ensuring that when gametes fuse during fertilization, the resulting offsprings have the correct full set of chromosomes. Genetic diversity. Meiosis generates genetic variation through two main mechanisms, that is crossing over. Homologous chromosome exchange genetic material, creating new combinations of genes on each chromosome. Independent assortment, the random orientation of homologous chromosome pairs during meiosis leads to the different combination of maternal and paternal chromosomes in the resulting gametes. Maintenance of chromosome number by creating haploid gametes, meiosis maintains a constant chromosome number for a species across generations, preventing the number from doubling with each generation. Evolution, the genetic variation introduced by meiosis is the raw material for natural selection, allowing population to adopt and evolve over time. I hope it is clear. Now discuss with your teacher about haploid insects and lower plants where cell division occurs. See first, haploid insects and lower plants where cell division occurs. These organisms, they are haplontic, means haploid life cycle, where the dominant life stage is haploid. For example, जैसे insects हैं, male honeybees, wasps, ants, and they are haploid and produce sperm through mitosis. Lower plants जैसे algae like Chlamydomonas, Spirogyra, mosses, they are example of haploid stage dominant. Haploid gametes are produced through mitosis, and the diploid zygote formed by the fusion of haploid gametes immediately undergoes meiosis to produce a new haploid organism. Second, some haploid cells in higher plants where cell division does not occur. Now see, some haploid cells in higher plants where cell division does not occur, for example, in higher plants which have a dominant diploid stage, some haploid cells are specialized and do not divide. They include as a synergids, cells in the embryo sac that help guide the pollen tube, antipodal cells, cells at the other end of the embryo sac from the egg cell. These cells are part of the haploid female gametophyte and perform a structural or supportive role rather than undergoing cell division to form a new organism. Other examples include mature sperm cell, that is spermatozoa, and egg cell, that is ova, which are produced from the diploid sporophyte but are haploid and do not divide further before fertilization. Right? Can there be mitosis without DNA replication in S phase? No, there can be no mitosis without DNA replication in the S phase because mitosis is an equational division that requires a duplicated set of chromosomes to produce two genetically identical daughter cells. DNA replication ensures each chromosome is duplicated, allowing the sister chromatid to be accurately separated during anaphase, thereby maintaining the same number of chromosomes in each new cell. Next question, can there be DNA replication without cell division? Yes, this we have done it. Yeah, yes, there can be a DNA replication without cell division known as endomitosis. This occurs when chromosomes replicate multiple times, but the cell's nucleus and cytoplasm do not divide, leading to cells with multiple copies of the same DNA. This process is observed in certain cells such as polytene chromosomes in the salivary glands of Drosophila. Next, analyze the events during every stage of cell cycle. Notice how the following two permanent: number of chromosomes and per cell, amount of DNA content C per cell. Now here see the first part. In G1 phase, the number of chromosomes N per cell remains same and each chromosome is formed of one chromatid. In S phase, each chromosome is formed of two sister chromatids joined at the centromere. Some condition continues in G2 phase, while in M phase, sister chromatids separate and move to different cells, but the number of chromosomes remains the same in mitosis. In second condition, in G1 phase, the amount of DNA content, C means content, per cell remains same, but in S phase, it is doubled due to DNA replication. It remains double in G2 phase, but is halved in M phase of cell cycle. I hope everything is clear. Any doubt anywhere, ask me in the comment section, but don't forget to subscribe the channel. Thank you. Again, we will meet in the next video.